is going to be my presentation on the protein Maltase Glucoamylase. So Maltase Glucoamylase is going to be coded by the MGAM gene, which is located on chromosome 7. And this protein is going to contain 1,857 amino acids. So the function of this protein is that it's an alternate pathway for starch digestion, and this is used when the body lacks alpha amylase. So this protein is going to play a special role in the digestion of oligosaccharides. It's going to be present in tissues such as the small intestine, the colon, and the kidney. And its target location is going to be the membrane of these intestinal cells. So there are 6 steps in the pathway of a protein. The first one is transcription. The second one is the arrival of at the rough endoplasmic reticulum. The third one is translation. The fourth one is arrival to the smooth endoplasmic reticulum. The fifth one is going to the Golgi apparatus. And the sixth one is reaching the cell membrane, which is where our protein will end up. So the first step is transcription. So this is going to be a process which occurs in the nucleus. And it's going to get started off when the DNA unravels itself at the location of a specific gene. So from there, the RNA polymerase, as you can see in this picture, it's going to come and then it's going to form a specific mRNA strand for that gene. So in our case, it's going to be the MGAM gene. Next, comprising of steps 2 through 4, we're going to have our mRNA go to the endoplasmic reticulum. And the rough endoplasmic reticulum contains lots of ribosomes, which are going to be used in protein synthesis. So at the ribosome, it's going to undergo a process called translation. So this is when tRNA molecules are going to bring amino acids to the ribosome based on the different mRNA codons that it reads. So all these amino acids are going to bind together and they're going to create a pre-prohormone, which is basically the precursor to a protein. So after translation is done, the rough endoplasmic reticulum is going to check for any defects in the protein. And if it finds any, it's going to destroy that protein right there. If the protein passes that step, then the rough ER is going to chop up signal sequences of the pre-prohormone, and it's just going to create a prohormone, which is now a protein. This prohormone is going to leave the rough endoplasmic reticulum and move on to the smooth one. And in the smooth portion of the endoplasmic reticulum, lipids are going to be added to the protein, and after that, the protein is going to be sent to the Golgi body. So now we're at steps 5 through 6, where the protein is going to be at the Golgi apparatus. The proteins travel to the Golgi apparatus using a vesicle. So a vesicle is basically a bubble-like structure used to transport cellular products. So the Golgi apparatus is going to manufacture, store, and transport these different proteins. It's also going to synthesize some sugars and add them to the proteins, as well as fold the proteins. So finally, the proteins are going to be stored in the Golgi apparatus and wait there till they're needed. And when they're finally needed, they're going to leave the Golgi body using vesicles. So they're going to enter the Golgi apparatus using vesicles, and they're going to leave it using vesicles as well. So Maltase Glucoamylase is going to end up on the cell membrane of the intestinal cell. So the vesicles don't really have to travel that far. They just need to make it to the outside of the cell membrane, where they'll stay there and they'll do their starch digestion. So these are some mutations that are going to be caused by the MGAM gene. Unfortunately, we don't know the specific genotypes and the locations of the gene where these mutations occur, but we do know all the phenotypes that are a result of these. So these include increased Vaccinia virus infection, the decreased viability of nitrite, shRNA abundance by approximately 50%, increased G1 stage DNA content, increased gamma H2AX phosphorylation, reduced mammosphere formation, decreased viability in ovarian lineage, and Maltase Glucoamylase is going to be used as a prognostic marker for renal cancer. So as your renal cancer progresses, the levels of Maltase Glucoamylase are going to decrease. So that's how doctors can diagnose what stage of renal cancer you're at. So this represents my protein Maltase Glucoamylase, and this is because that protein is very folded. So these party decorations and this string represents the multiple layers of folding this protein has gone through. And I also added some golden stars at the bottom of this protein, as well as the top portion over here. And this is because maltase glucoamylase has 2 binding sites, and these golden stars represent the binding sites of the protein. Page for the presentation. And these are all the image citations I had.